All right, so let's pick up where we left off, the master genetic code. First of all, there is a structure called a codon, which are groups of 3 nucleotides that dictate which amino acid is added to the growing peptide chain. There are 64 different triplet codes from 20 amino acids. There are some amino acids that are represented by several codons. This is called redundancy, which will allow for the insertion of correct amino acids even when mistakes occur in the DNA sequence. A phenomenon known as wobble is how these things actually attach. It's actually only the first 2 nucleotides that are required to encode the correct amino acid. And the 3rd nucleotide doesn't change the sense of the codon. So if you have a mutation, and we'll talk about mutations here in a little while, but if you have a mutation that only involves that 3rd letter, in the codon, the 3rd nucleotide in the codon, the correct codon is still going to get attached, and so that's not going to change our genetic information. So that 3rd codon, while it must be there for the process to work, it's not involved in the actual attachment of that of that amino acid. Therefore, that is referred to as wobble, because the new DNA sort of hooks and then sort of swings down into place like that. This wobble will prevent certain variation or mutations without altering the message. And again, later on, we will talk about mutations and the role that they play in certain things. Still not happy with my camera angle here. That's a little bit better. All right, these are the codons. I'm not going to ask you to remember all of these codons, so rest assured, you don't have to remember all 64 of them. I do want you to remember several of them, however. I want you to remember that AUG, which is the codon for methionine, is also referred to as the start codon. This one is a really easy one to remember for me because the school year starts in August. The 3-letter abbreviation for the month of August is AUG. There are 3 other codons that I will ask you to remember specifically, and that is UAA, UAG, and UGA. Those are all referred to as stop codons. University of Auburn, Alabama, or Alabama at Auburn. I know the technical name for the college is Auburn University, but I can remember UAA by University of Alabama at Auburn, the University of Athens, Georgia, and the University of Georgia at Athens. I can remember that. And again, I am a college football fan, and that's how I remember those 3 stop codons: UAA, UAG, and UGA. Those are the 3 stop codons. So for the process of replication or transcription to start, requires this codon, and for transcription to stop, requires one of these 3 codons. The start codon is always AUG, but it does code for a different form of methionine. Messenger RNA encodes for several genes in the series. Eukaryote only codes for 1 protein. For transcription and translation, occur simultaneously in the cytoplasm, and this is what's going on in bacteria, by the way. This is a eukaryote. Transcription occurs in the nucleus. Translation occurs in the cytoplasm. Genes exist as an uninterrupted set of triplet coding for a protein within bacteria. Genes contain introns that don't code for anything, exons that do code for things. The introns will eventually be edited out. Hopefully in your biology class you went through the process of intron excitation and being removed from the product. There are certain control mechanisms that ensure that genes are active only when they are required. These are enzymes. They are produced as needed. The most common referred to and talked about one of these regulatory systems and control mechanisms is something called the lac operon, which we will talk about in just a few minutes. Regulation in bacteria, archaea, and eukaryotes. Antisense RNAs will actually stop the production of RNA. It's the antisense RNA makes it. Antisense stops it from being made. MicroRNAs also play a role in the control and regulation of bacteria, as do riboswitches. Operons, again, which we'll talk about one specifically here in just a moment, are only found in bacteria and archaea. They consist of a coordinated set of genes regulated as a single unit, reproducible and repressible. In other words, they can be stimulated to start and run quickly, or they can be stimulated to stop and not function at all. Catabolic operons are induced by the substrate of the enzymes for which they are structural gene, or for which the structural genes codes. And remember, catabolic means you're breaking things down. They only produce the enzyme when that substrate is present. The lac operon, the one we're going to talk about, is this type of operon because it takes the lactose to be present for this enzyme lactase to be made. There are repressible operons. These are anabolic operons or anabolic enzymes. Again, anabolic means we're putting things together, making bigger things out of smaller things. These are turned off by the product synthesized by the enzyme. The lactose operon, knew we'd be there, is a model for an inducible gene regulation in bacteria. The one big mistake students make in the beginning is when they see the word lac operon, they think this is the way that human beings process lactose, and this is not. You will notice the lac operon is a regulatory mechanism in bacteria. This is a system where some organisms, some bacteria can metabolize the lactose sugar in milks and some cannot. And this is how they do it, using this lac operon. It is a regulator. It's composed of a gene that codes for a protein capable of repressing the operon. That gene capable of repressing is called a repressor. The control loci or locus has a promoter, which is recognized by the RNA polymerase, and an operator, which is the on-off switch. There is a structural locus that is made up of 3 genes that codes for different enzymes needed to catabolize lactose. And here is the lactose operon. Again, the person who made these slides do not work very well in a lit room. But there's the regulator. You see it's not attached here. There's the promoter, there's the operator, and there's the genes moving forward. That's the, hang on, let me see if I can read that. That is the repressor protein. That's the regulator of the repressor protein. You see it's there, and nothing is happening. Here you get the lactose, the DNA polymerase attaches itself on the promoter. As it moves, it knocks this out of the way, and things are allowed to move on. This will eventually come back in and close the system down. Provided there's plenty of this available, this will remain unattached, and this process can go forward. As this gets used up, eventually these will be able to bind themselves and stop the production of those enzymes. When bacteria turn off or on a complement of genes that leads to obvious phenotypic changes, that is referred to as phase variation. These phase variation genes are heritable. They are mediated by regulatory proteins found within the cell, and they most often apply to traits affecting the surface of bacterial cells. A couple of examples are Neisseria gonorrhoeae, the causative agent of gonorrhea, and Streptococcus pneumoniae, one of the causes of pneumonia. Now, here's the thing. In its normal phase, Neisseria gonorrhoeae and Streptococcus pneumoniae are not pathogenic to human beings. It is only when they are in the phase where the gonorrhea is producing attachment fimbriae and Streptococcus pneumoniae are producing a capsule that they become pathogenic. So one phase, they're not pathogenic, but in the phase variation where they have inherited the proper genes to make a attachment membrane, then this then can attach itself to the inside of the human organ, primarily in the urinary tract, and then develop this disease. This one produces a capsule, so when these bacteria get into the lungs, because of that capsule formation, it delays the body's response, the body's immune response to those, and then we can get pneumonia. This one, it's because it's in an area of the urinary system, the interior of the urinary system, the urethra, the bladder. There is not a whole lot of immune response that goes on there. That's one of the reasons humans are so susceptible to urinary tract infections, because there's simply not a lot of immune response. There's some, but not a lot that happens there. And when these bacteria are allowed to attach themselves on the inside of our urinary tract, they can very quickly build up and form this disease. DNA recombination events, simply put, recombination is where DNA is put together in different patterns. So it's being recombined. Bacteria have no exact equivalent to sexual reproduction. An event in which one bacteria donates DNA to another is referred to as recombination. The end result of that new strain of bacteria, or the different form in both the donor and the original recipients. So bacteria A gives a little piece of its DNA to bacteria B. Bacteria B gives a corresponding piece of its DNA to bacteria A. The offspring of B and A will now be C and D, both different from A and B. The one that A produces will be different from A, but it's also different from B. And the one that B produces will be different from B, but it would also be different from A and C. Hopefully that made sense. But by doing that, we get a lot of recombination, we get a lot of variation in bacterial species. This depends on the fact that bacteria have plasmids and are very adept in interchanging genes. Bacteria can provide genes for resistance, or this process can provide genes for resistance to drugs and metabolic poisons. It can make them capable of using new nutritional and metabolic items, and it can increase the virulence and the adaptation to the environment. A recombinant is any organism that contains genes that originated in another organism. So A, again, got a little bit of DNA from B, then its offspring C would be considered the recombinant. Any transfer of DNA that results in an organism acquiring new genes that did not come from its parent is referred to as horizontal gene transfer. Plasmids are small circular pieces of DNA. They contain their own origin of replication. In other words, they can replicate independent of the cell. They are not necessary for survival. Many of them carry useful traits and give the bacteria that they are found in abilities that they would not have if it were not for the plasmid. There are also structures called chromosomal fragments, which must be integrated into the bacterial chromosome in order to be replicated. So while the plasmid is not bound to the replication of the cell, chromosomal fragments are. Plasmids can replicate independently. Chromosomal fragments must replicate themselves as the cell itself is replicating. Different types of horizontal gene transfer. There's conjugation. Again, not going to go through all of these, but these are the different types. Transformation and transduction. Some of the things that you need to point out here, that in transduction, the donor cell is lysed. So this bacteria cell dies, whereas transformation and conjugation, the donor cells don't necessarily die as a result of the transfer. Exchange of genes in conjugation. First of all, the fertility pili, the F factor, is a way that allows for synthesis of conjugation. Conjugation is a mode of genetic exchange whereby a plasmid or some other genetic material is transferred by a donor to a recipient via some sort of direct connection. Most common way in gram-negative conjugation is through a structure called the fertility or F factor pili. The recipient cell has a recognition site on its surface. The F plus cell has the plasmid. The F minus cell lacks the plasmid. Contact is made when a pili grows out of the F plus cell, attaches itself to the surface of the F minus cell, contracts and draws the two cells together, and then in a gram positive, an opening will be created between two adjacent cells. The replicated DNA then passes through one cell to another. So in conjugation, for gram negatives, the fertility or F factor pili is required. For gram positive conjugation, no such pili is required. Conjugation is a conservative process in which the donor bacteria retains or conserves a copy of the genetic material being transferred. The F factor in E. coli, for instance, again, E. coli is a gram-negative rod that we talk about a lot in lab. The donor, or the F positive cell, makes a copy of its F factor, or its little plasmid, transmits that to the recipient, or the F minus cell, through the structure of that pili, which we just talked about. The F minus cell is then converted into an F plus cell, which will be capable of producing a pili and conjugating with other cells, thus spreading that ability throughout a population or a colony of E. coli. High-frequency recombination donors, or HFRs, is where a plasmid becomes integrated into the F plus donor chromosome. When that is replicated, it begins to transfer to the recipient cell. Some of these chromosomal genes will get transferred to the recipient, and the plasmid genes may or may not be transferred. Resistance plasmids, or resistance factors, can be found in bacteria, and they bear genes for resisting antibiotics. They are commonly shared among bacteria through conjugation. They can pass on one or more of the following characteristics: either resistance to multiple antibiotics, resistance to heavy metals, or synthesizing virulence factors such as toxins, enzymes, and adhesion molecules. This, of all the slides we need to be concerned with as far as medical ramifications, this is it, and the way resistance plasmids work. This is how organisms develop, or bacteria develop, resistance to our drugs. This is why things like taking your antibiotics the way they are prescribed and for the length of time they are prescribed are so very important. Transformation is when a chromosome is released by a lysed cell, breaks into fragments small enough to be accepted by a recipient cell. That is referred to as transformation. It is the non-specific acceptance by a bacterial cell of small fragments of DNA from the environment. It is facilitated by DNA binding proteins in the cell wall, and the cell must be competent, is the term we use. That means the cell is capable of accepting genetic material. That cell is considered to be transformation competent. There are no special appendages required. Donor and recipient cells don't have to be in direct contact. These small fragments of DNA are just released into the environment where these organisms live, and if the donor cell has the proper attachment sites, they can pick this material up. Foreign genes will form completely unrelated organism, or from a completely unrelated organism, are inserted into a plasmid. That plasmid is then introduced into a competent bacterial cell through transformation. This type of thing can be carried out in a test tube. Many of the things we do to make new medicines, new drugs, new products are done with this type of transformation process. Transfection is a similar process carried out in eukaryotic cells that form genetically engineered yeasts, plants, and mice. When we talk about later on in the semester, we talk about goats, that are producing human vitamin, human genes in their milk. That is, they were created as a product of transfection. Generalized transduction is where a bacteriophage serves as a carrier from a donor cell to a recipient cell. There are random fragments of disintegrating host DNA are taken up by the bacteriophage. Virtually any gene from a bacterium can be transmitted through generalized transduction. Specialized transduction is where very specific parts of the host genome is incorporated into the virus. It is explained by a prior existence of a temperate prophage, which was inserted into a fixed site of the bacterial chromosome. When these are activated, the prophage DNA separates from the bacterial chromosome, carrying a small segment of host genes with it. During the lytic cycle, specific viral host gene combinations are incorporated into the viral particles and carried to another bacterial cell. So basically, generalized transduction, just any piece of DNA can be picked up and taken into the bacteriophage. Specialized transduction, very specific parts of the DNA have to be picked up. Transposons, or jumping genes, are transposable elements capable of moving from one part of the genome to another. They can be transferred from a chromosome to a plasmid or vice versa, or from one cell to another in bacteria and in some eukaryotes. Some will replicate themselves before jumping to the next location. Some simply move. These are involved in changing traits such as colony morphology, pigmentation, and antigenic characteristics. Maybe you will see the jumping gene could be the result of something that should be producing a small white colony, converting it into larger yellow colonies. These can replace damaged DNA, and they can also play a role in intermicrobial transfer of drug resistance in bacteria. Again, here's the transposon. Here's the transposon. You see it's here, and now it's jumped over to here. Here it's jumped into several different places, and here it's actually jumped out of the bacterial chromosome and into the little plasmid. So several different routes that a jumping gene can do as this cell is producing offspring. We're going to stop there for the moment. Let me give my voice a rest, and I will finish up this chapter in a few minutes.